Welcome to NewYorkBIO's Patient Engagement Summit, an annual event bringing together patients, advocacy groups, life science, and biotechnology leaders. Our next panel is on the future of biomarkers, featuring Michael Davoli from ACS CAN, Dr. Wojciech Swat from BioReference Laboratories, Dr. Carl Morrison from the Roswell Park Cancer Institute, and Tiffany Westrich Robertson from AI Arthritis.
Good morning, everyone, or I should say good afternoon. It is afternoon. I'm so excited to be here with all of you today. My name is Michael Davoli. I am the Senior Director of Government Relations for the American Cancer Society Cancer Action Network, ACS CAN. We are so excited to be joined by so many partners to talk about the importance of biomarkers and biomarker testing. We here, I just want to say at ACS CAN, we often love to talk about the future of cancer care. And to us, biomarker testing truly is at the forefront of patient care. Not just cancer, but all sorts of other chronic diseases and illnesses. So we have an incredible panel lined up today and for today's discussion, and rather me just sort of reading through these incredible bios for each of our panelists, I'm going to encourage everyone to, as soon as we're done here, to take a few minutes and look at their bios on the website for the summit and read a little bit about them. And but first, what I want to do is get right into the meat of this. So we've got three wonderful panelists. First, Dr. Carl Morrison, who is the Senior Vice President of Scientific Development and Integrated Medicine at Roswell Park Cancer Institute. I've had the pleasure of working with Dr. Morrison now for more than a year on these biomarker issues. Second, we have Dr. Wojciech Swat, who is the Interim Director of Cancer Genomics at BioReference. And finally, we have Tiffany Westrich Robertson. Tiffany is the CEO of at the International Foundation of Autoimmune and Anti-Inflammatory Arthritis, AI Arthritis, and she's also a patient living with AI arthritis. So we are incredibly excited to have all of you here. So why don't we get right into this? I'm going to go to you, Dr. Morrison, if I could. Tell us about biomarker testing. Pretend we're all brand new here. What is biomarker testing and why is it so important to precision medicine?
Well, the definition of precision medicine is that you know something unique about that patient, right? Something unique that's causing their disease. And that something that is unique is typically referred to as a biomarker. Now, more specifically, a biomarker means that, a certain element of that patient's body, whether it's DNA, RNA, proteins, has a unique change that in some way we can measure. And when we measure that unique change and report it, we refer to that as a biomarker. And, different drugs have been developed that specifically focus on one biomarker, and that is really the definition of precision medicine.
So thinking about this a little further, just to dig into this, Dr. Swat, can you give us some examples of how biomarker testing is actually being currently used in oncology, whether it's lung cancer, breast cancer? I've heard a lot of acronyms, but is there, can you give us some examples of how it's actually being used?
Absolutely, Michael. Thank you very much. I will go straight into my own field of cancer genomics, and I will use some examples that are molecular biomarkers. And with those, we can use them at diagnosis to sometimes refine diagnostic criteria of diseases, be that solid tumors or cases with blood tumors, hematological tumors, where the molecular analysis is becoming increasingly relevant for sometimes it's disease defining and strictly diagnostic, such as presence of BCR-ABL fusion in CML. And in many other cases, it can be very helpful to hematopathologists and solid tumor pathologists in trying to make correct calls as far as the tissue of origin, the cell of origin in the tumors, also known as tumor primaries in case of metastatic tumors. So there's a very important diagnostic role for biomarkers. There's also a hugely relevant role for biomarkers in making prognostic calls. And in some cases also predictive. This is something where this is the new era where we can actually predict across a certain tumors based on the constellation of molecular markers. And I'll give you some examples of that. Obviously, there are therapeutic implications, and this is a growing list of now FDA-approved drugs which are used in patients based on presence of specific molecular lesions. This is obviously extremely relevant and can be life-saving. The example of CML is probably unique, where the drugs, the STI-341, also known as Gleevec, have essentially completely changed the landscape and making this a chronic disease or curable disease, which was previously a killer. And there are many examples of solid tumors of newer drugs, such as sotorasib in the treatment of G12C KRAS containing variants in non-small cell lung cancer, and many other examples. But there are still, it's a growing list, but it is a limited list. And I will tell you why it's important to keep this in mind. And I also wanted to lastly point out to something that's becoming increasingly critical, which is the follow-up testing in the context of MRD, or measurable residual disease, also known as minimal residual disease, that is becoming prevalent and really critical in assessing the therapeutic response, but also monitoring for refractory and relapsed features of neoplastic disorders. And let me just, if I can use another 10 seconds to sort of define a little bit better of what I'm talking about. So in my field of cancer genomics, these biomarkers would constitute of single nucleotide variants, such as KRAS G12C. It's a single base in your 6 billion genome that is mutated, giving rise to a somatic or tumor-only change that becomes a tumor driver and makes these tumors grow uncontrollably. Those can be gene rearrangements or fusions, such as BCR-ABL I mentioned, or such as rearrangements of ALK or ROS1 prevalent in lung cancer. Those could also be gene amplifications, such as HER2/neu amplification or amplification of MET gene in lung cancer. HER2/neu being prevalent in many tumors, but also in primarily in breast cancer. Finally, there are some complex biomarkers that are emerging, where we don't completely understand the exact therapeutic and predictive utility, but because we integrate them into our testing algorithms, we are rapidly gathering data that will permit us to utilize them fully, such as tumor mutational burden, or TMB, which is essentially a number of tumor mutations per megabase of your genomic DNA. Also microsatellite instability, or MSI. Those are two biomarkers critical in stratifying patients for immunotherapies, such as immune checkpoint inhibitor therapies, which can be life-saving and are prevalent in clinical trials of tumors such as colon, lung, and melanoma. Lastly, there are really emerging tumor markers, complex tumor biomarkers, such as homologous recombination deficiency score, also known as SCAR or SCAR. This is a newer marker, HRD, which is gaining prevalence as something that can stratify patients for PARP inhibitor therapy, something that patients with prostatic, breast, and gynecologic tumors, primarily high-grade serous ovarian tumors, can benefit tremendously, something that can complement chemotherapy at very low level of toxicity. Thank you.
Thank you so much, Dr. Swat. So it's fascinating. Clearly there's so much happening there, and there's so many uses of this. But I gotta ask, I gotta go backwards for a second. So, Dr. Morrison, if you could, I'm confused. Genetic testing versus biomarker testing. What's the difference in the two? Because we're talking about biomarker testing, but I think everyone's heard of genetic testing. So can you just quickly explain the difference there, Dr. Morrison?
I think biomarker testing is a more inclusive term of all of our testing of biomarkers, but people use the terminology genetic testing in different ways. But I think the majority of people, when they talk about genetic testing, they're talking about germline testing, right? What you might inherit from one of your parents, right? What is your risk of having breast cancer when you're 40 years old, or colon cancer when you're 45 years old, right? And the comparative term when you're not talking about genetic testing, we usually think of somatic mutation testing, but all those together are biomarker testing. They all fit various pathways of the treatment plan, whether you actually have cancer, whether you're at risk of cancer. Biomarker testing includes all of those, like BRCA1 and BRCA2 are really famous for germline genetic testing for patients at risk for breast cancer.
Thank you so much, Dr. Morrison, for that clarification. So Tiffany, I'd like to go over to you if we could. You've heard the other panelists introduce biomarkers, talk about the differences between genetics and genetic testing and biomarkers, and some of the practical examples. So this is now becoming really a key part of the immunology world. I always have a hard time saying that. Tell us a little bit from an organization and from a patient perspective, more about the importance of this shift.
Sure, thank you. Well, so immunology in regards to precision medicine and biomarkers is relatively in its infancy. Well, okay, or let's just say in early adolescence. So today I'm going to take a step back a little bit too and talk about the research that's underway regarding biomarkers in autoimmune, autoinflammatory arthritis diseases like rheumatoid arthritis, lupus, for example, before jumping into biomarker testing in the clinical setting. So first, why is the shift into the immunology space important and frankly really exciting, I want to add. So the first point is that our diseases are heterogeneous, which means that the disease presentation and progression varies per individual uniqueness. It's really important to know because that really can interfere with diagnosis, and the individuality happens even within the same diagnosis as well. So that also means that most of us don't tick those boxes. We don't match what the textbook disease definition may say, and that could lend to delay in diagnosis. So first, identifying specific disease biomarkers can help expedite the diagnostic process. So then this can lead to a quicker access to the right treatment for the right time based on a patient's unique presentation. And since we are so diverse, even with several treatments on market, what works for me may not work for the next person. So this is important to note because biomarkers could be that key to finding the right treatment at the right time in the disease journey. And this is huge for early disease because there's just a small window of time from onset where access to the appropriate disease modifying therapies is really vital. And delay in detection and diagnosis, that can lead to the delay in treatment. If the treatment isn't the right fit for the person, then the inflammation is uncontrolled, and that can lead to poor quality of life, chronic illness for life without the chance of ever achieving remission, comorbidities, and potential irreversible damage and disability. So finally, there are some diseases where biomarkers in our community are already identified, and current research suggests testing for them can improve outcomes and in some cases significantly increase a patient's chance for remission. And I have to say that again, that remission, because it is honestly a word that patients, we do not hear much currently in our diseases. These are typically lifelong chronic illnesses. And therefore, this is where we are in the shift and why it's so important to rheumatology and the immunology community.
Thank you so much, Tiffany. And so something you said there was really interesting about the timing of biomarker testing. And something I've often heard mentioned is that with precision medicine, it's about getting the right medicine at the right time to the right patient and stuff. So that sort of echoes kind of what you're saying there. And Dr. Morrison, I'm curious, in the clinical setting, what does it look like? What does biomarker testing really look like? How you administer in the clinical setting? In other words, is a patient's diagnosis and treatment journey, is the biomarker testing used just for like in the very beginning, or can it be used for someone, a patient who's been, undergoing treatment for a while? Tell us a little bit about what that experience is like in the clinical setting for a patient.
I think there's a certain degree of variability. It's related to the type of cancer that someone has. For like instance, lung cancer or melanoma, people instantly think about biomarker testing, right? If you have more obscure tumor types like what's called soft tissue sarcoma, people don't immediately jump to biomarker testing. And part of the reason why is because of the the number of precision methods that have been developed across the different tumor types. And certainly lung cancer, melanoma, breast cancer, they lead the pathway in precision medicine. But there is the other variability too that's dependent upon, probably more precision biomarker testing done at academic medical centers than there are in community settings. Various reasons for that. Some of them's education. And then there's, many other things like what's the stage of the disease. Certainly, there's more biomarker testing done for advanced stage disease than for early stage disease. So there's a lot of variability, but a lot of the variability is just knowing the technical and clinical aspects of when you should do biomarker testing. And there's a certain degree of education that's required to really understand the process. And it's a gap sometimes in some communities.
Thank you so much, Dr. Morrison. Now we have a question from the viewers here, so I'm going to ask this. So maybe I'm not sure who's either Dr. Swat or Dr. Morrison. Are we ever going to move toward a more universal diagnostic or biomarker regimen reliant on the whole genome sequencing as opposed to gene panels or other modulates? So, what are the barriers to this? And I know we're going to talk a little bit about like insurance barriers and stuff, but I don't know if either of you want to take a stab at that question.
I could take this if that's okay, Dr. Morrison.
Fine, sir.
There's with the advent of genomic testing, which is now becoming increasingly feasible due to the massively dropping costs of genome sequencing, both DNA and RNA. There is an opportunity to test at the whole genome level for both DNA and RNA. Is this appropriate? In some cases, maybe. In some cases, may not be. There is a little bit of confusion regarding different modalities of genetic and genomic testing, such as screening versus diagnostic testing versus risk assessment. So, for example, if you get the 23andMe test, you are not being screened for cancer. You are doing a risk assessment. So those are important considerations that I think should be part of the discussion with patient groups so that they understand those testing modalities and can use them to their advantage. Let me give you an example of genomic versus non-genomic biomarker in something like immune checkpoint inhibitor therapy, the immunotherapy for cancer. The TMB that I mentioned, tumor mutational burden, is a genomic biomarker that's based on sequencing large genomic space to adjudicate the level of mutational activity in the tumor. A non-genomic marker equivalent for that would be staining for PD-L1, which is a regulatory protein that can be blocked on the surface of T cells and unleash them in lieu of essentially endogenous tumor vaccine. So those are pairwise biomarker usage examples that use completely different technological platforms, not next generation sequencing or NGS, or IHC, essentially conventional staining. I will also say that in my practice, we tend to provide our clients, the pathologists and oncologists, with the opportunity to have access to tumor-specific genomic panels. We routinely sequence, let me give you a specific example. We routinely sequence 523 whole genes in lung cancer patient specimens. However, we report only a slice of those genes in order to focus on those genes which are clinically actionable and not to overcrowd the clinician and the patient with genomic information that at this time may be superfluous. Our reports tend to be very succinct. We put all the relevant information that in lung cancer includes literally 7 genes that are relevant for FDA, include therapeutics, and maybe additional 20 which have NCCN, National Comprehensive Cancer Network, recommendations in their guidelines. And that's it. And our report with all the technical information and disclaimers fits on 2 to 3 pages. If we wanted to report the full panel, or God forbid the whole genome, our report would be 50 pages plus. With the workflow that clinicians such as Dr. Morrison experience in their clinic, this would essentially become a showstopper. So I think it's very important to realize that with the flux of genomic information at relatively affordable price, we still have to pay attention to what really is the limiting factor in communicating clinically actionable data, which is proper annotation of genes for diagnostic and potentially therapeutic, prognostic, predictive factors. And I can explore, I can expand on this if you would like to. But maybe one more comment about testing at diagnosis versus follow-up, or in direct response to your question, when to test. Any kind of MRD approach, again, measurable residual disease or minimal residual disease testing as a follow-up, generally requires a paired analysis at diagnosis. That is really the best way in which we can follow up what's going on with a cancer evolving and acquiring new somatic mutations and losing some, but also in autoimmune diseases, where we can follow offending clones in real time in the follow-up. Once we know who is the bad guy, which is best adjudicated at diagnosis. Thank you.
So thank you so much, Dr. Swat. And I want to shift over to Tiffany. I've got a number of questions actually coming into the chat box for you, but I want to start off by this, at the American Cancer Society, obviously our focus is cancer, but I think one of the things that really excites us and others about this issue is this is something that goes way beyond cancer. It's like strike the word cancer from any documents around biomarker testing, because this impacts patients of all different stripes from all different chronic diseases and illnesses. So as a person representing the immunology community, I'll get that word right before the end of today, I promise. Specifically the AI arthritis community, what does the promise of biomarker testing mean to you? And then I do have a follow-up question from the audience after that.
Okay, great. Well, first, I am going to answer it from a patient perspective and try to sort of capture, I guess, the perspective of many. And I'm going to say it means that those with new onset may never know what it means to be that mystery patient like I was for years before getting a diagnosis. It can mean that we may have a chance to continue playing sports or working a regular 9-to-5 job or contributing to society to the degree that we would like to. It means that more people may never realize what it feels like to constantly cancel on family and friends because they're too fatigued or they're too in pain to be there. And I should also point out that the average onset for AI arthritis diseases is 20 to 40 in adults, and then it also in children. So this means that a lot of us are in that prime of our lives. And it means that more people will get the treatment that they need to avoid disability and remain in the workforce or go to school and really be a productive part of society. So it just means that there's a chance that more patients will achieve remission instead of living the rest of their lives with chronic illness. And it also means a more balanced healthcare system because the overall costs are going to be less.
So following up on that, and this is a question from the audience, do you think it's that, how much of it is that doctors are not using biomarker testing, when they could be? And if that's the case, how often do you think it's really because of cost, or is it because they're just sort of not aware of the latest in medicine?
That's a great question. So maybe a little bit of both. Our organization attends every year the American College of Rheumatology Convergence meeting, and then also EULAR Congress, which are the two biggest rheumatology scientific community conferences annually in the world. And I can tell you, it's only been the last couple of years that precision medicine, and they didn't even use the word precision medicine until this year. We were like, oh, see, we told you it was coming. And so the research is so early and so new, even though, for example, in rheumatoid arthritis, it's known that elevated ACPA and rheumatoid factor positive people will tend to have more aggressive disease, and there's widely clinical availability to test those. So in those type of situations where the research has really been underway, there's also some novel biomarkers in lupus. I would say that depending on what your condition is, it would be more of a conversation. So is that up to you as a patient to know, or the doctor to know? I think it's really up for all of us to keep up to date. And I know I can tell you at AI Arthritis, we're definitely doing that, with hopefully helping patients to be able to start that conversation. So it's going to depend on your disease, and then if you have cancer or some of the other chronic conditions that we've been talking about, I think it really depends on how far we are and how available. So then once we get to the availability, then we'll run into the loops of insurance and those type of barriers.
Well, thank you for that. So Dr. Morrison, I actually would love to go back. Before I ask you the question, if you wouldn't mind, you mentioned to me on a previous discussion a brief story about a patient that you're working with or worked with at one point who used biomarker testing later in their diagnosis. I think they were undergoing treatment. Would you mind just giving just a very quick summary of that story? Because I think that was really fascinating.
I think you're probably referring to I previously told you that when we first started down the journey of biomarker testing and precision medicine, it was 10, 12, 15 years ago. We were bringing up the first test in our laboratory, and of the first 40 patients we tested, one had a very unique biomarker that had a targeted therapy or for precision medicine. And while this wasn't at that time an official test, right, we still saw the result. And I reached out to the IRB and got approval to contact the treating physician and tell him of this result. Now, at that point, I knew nothing about the patient, zero. And the short of the story was, this was a 50-year-old lady who had previously been treated with several rounds of chemotherapy, lung cancer, several rounds of chemotherapy, radiation therapy, failed everything. She was on hospice. They'd given her only a few more months to live at most. And they started her on the precision medicine. And 3 years later, she ran in a marathon. 10 years later, she's very thankful that she lived long enough to see her two daughters both have children and grandchildren. We fast forward today, and she has had a recurrence of her disease, but she had 10 or 12 years of really good life, and she's so thankful that she got to grow up and see her grandkids and have time to tell everybody, to live her life to a little bit fuller extent and appreciate it.
Incredible. Thank you so much, Dr. Morrison. And look, I think that's kind of summarizes why we're doing this, just to try to give people a chance to live their lives, right? And so biomarker testing, it's right, it's only as good as it is accessible. Are there different, and this is for you, Dr. Morrison, are there different populations that are less likely to get access to biomarker testing? I mean, are there disparities, whether it's rural versus urban, rich versus poor, racial, anything like that?
Yes, there are, and there have been studies out there to really show that. I think probably the most cited study is the one that came out of Dana-Farber in 2019, where they used Medicare claims data to show that the rate of biomarker testing in Blacks was essentially 1/2 that of the white Caucasians, right? They also went on and studied to show, that poverty has an impact upon this. I don't think anything of this is surprising, right? Mm-hmm. Then there's the other side of this. That's one barrier to testing, is that, many disparate populations have been actually left out of the original testing that developed the drugs for precision medicine. So that, if you go back to, where did a lot of this information come from? And a lot of it originally came out of the what was called the Cancer Genome Atlas study, TCGA, probably the largest national study we ever did on cancer genetics. And if you look at that population, it was 80% white, right? And it was like 5% African American. So, there was, sometimes unique populations can have unique markers. And if you don't test enough of them, you'll never find the marker, and you'll never develop the precision medicine. So there's all kinds of inequities that occur throughout the process of biomarker testing.
Thank you for that. And that does really echo the importance of the work that we're doing today, and as in the advocacy community, and mobilizing patients and stuff, because you could have these fantastic tests, but if they're not available to people, then how good really are they, right? And so that's our job in the advocacy space, is to be fighting for these things. So Dr. Swat, I'm going to put the question to you, which is the question that I get on a regular basis from lawmakers. This sounds like it costs a lot of money. I don't know about this, like, this is going to cost us a ton, and we can't really afford this. So tell me about, realistically, how cost effective is this, in the overall treatment of patients?
Thank you. So speaking of biomarkers in general, they range from very inexpensive. Let me give you an example of hemoglobin AC1 testing in assessing the level of necessary intervention in type 2 diabetes. It would be an example of an incredibly inexpensive test. Now, in cancer genomics in particular, the testing tends to be a little bit more expensive, as it involves next generation sequencing, NGS. But we are now really beating those costs with the availability of new testing platforms. And for most critical cancer genomics assays that are DNA or RNA based, we can cap the cost within a few $100s. And the return on this investment is incredible. If we can test every patient with secretory breast carcinoma for the presence of NTRK fusion, we will find that 9 out of 10 actually have that fusion, and we can treat them with latrectinib and probably save their life, which is quite incredible. I would like to allude to what Tiffany was saying about testing in especially autoreactive and immune disorders. There is a pretty good overlap in the lymphoproliferative disorders between disorders which affect, which are reactive, essentially autoimmune disorders, and those that tend to cross the line towards neoplastic process. And molecular testing, such as in the field of clonality testing, where we molecularly adjudicate the repertoire, the complexity of the repertoire of the responding cells, or neoplastically growing cells, can literally give a digital yes or no answer to some of those questions that affect patient management. So those tests can be incredibly accurate in distinguishing psoriatic lesion from a cutaneous T-cell lymphoma, something that would have dramatically different clinical course and potential for therapeutic intervention. Those tests can now be applied again with costs that can be reasonably capped at hundreds, if not few $1,000s. Patients with multiple myeloma can benefit from MRD by molecular testing of their clonality status. And you have to juxtapose that with very expensive testing modalities involving scanning with radioactivity, where the costs will be in the order of many thousands of dollars, and the molecular testing can be more accurate, quantitative, and actually much less expensive. Testing of PD-L1 by IHC is inexpensive, and it's a very good biomarker for stratifying patients for immune checkpoint inhibitor therapy. Thank you.
Thank you so much. And we got a comment in the chat box to me, not so much of a question, and it was fascinating. It is true. It's the real question is, what is the cost of a misdiagnosis, or what is the cost of using a treatment that is not the most effective treatment? And it's like, this is not. Someone said to me one time, this isn't your grandparents' cancer treatment. Right? Like precision medicine is what we're talking about, and biomarkers is sort of unlocks that precision medicine. So going back to Tiffany, because we're winding down here, as biomarker testing and starts, entering this clinical space, what do you suggest patients, patients and their loved ones learn about biomarkers, and what questions should they be asking their doctors, their healthcare providers, in order to ensure that they are getting that access to biomarker testing?
Yeah, sure. Well, first, I do want to just add a comment to the prior conversation and regarding the access, because it is important for patients and their families to stay educated on this, not just so that we can learn potentially better ways to treat our own diseases and go into remission, which is obviously a goal for families and patients, but we have to remember to use our voices. And it's important to talk to legislators to make sure that they understand that the value that's going to come from access to these tests and to precision medicine treatments, because it does cost a lot of money to keep failing and starting over because the insurance company decided the best treatment for us instead of that decision making between the patient and their physician. So understanding it, and there's lots of ways you can get involved with being a voice for the importance, because that is what's going to help move the needle for the access that we're talking about is a barrier that we all know. So as far as questions, it depends on the person. Not everybody walks into the doctor's office like me and has no problem talking whatsoever. So, we all have our different behaviors, our different security, confidence levels. I think just the basic question: are there biomarkers known in association with my disease that we can test for? That one simple question can open the dialogue if the doctor isn't already going to bring that up. So I do think that having the conversation, educating yourself as much as you can, reach out to whatever that organization is for your diseases. Obviously, if you have an autoimmune or autoinflammatory arthritis disease, we are very, very invested in this space, but there's other organizations as well. So work with those patient organizations to get educated on the topic and have a voice if you can. And then when it comes to your doctor, just don't be scared to ask that one simple question, and then go from there with the conversation. But I think also just asking them, obviously, about access, we just can't get away from that. And maybe even calling your insurance company if you are insured ahead of time and being prepared for that conversation and seeing if your doctor has alternative methods of gaining access to those tests.
Excellent. Thank you so much for that, Tiffany. And so before we wrap up, I want to make a plug to something that Tiffany said about reaching out to advocacy organizations. We've kind of danced around this a bit about how do we increase access to biomarker testing for all diseases and chronic illness. And it starts with legislature, starts with lawmakers getting engaged in this and passing legislation to expand access to biomarker testing. And we here at the Cancer Society are working with patient advocacy groups and provider groups across the state of New York to build a robust coalition in support of legislation that would increase access to biomarker testing. And we're inviting everyone to sort of be a part of this campaign. There's legislation that was introduced in the New York State Senate and the New York State Assembly earlier this year. We're going to be building support for that legislation. We're going to be fighting hard for it. But in order for us to be successful, we need you. We need all of you. We need your patient stories. We need your organization support. We need, doctors. And so I just want to leave it at this: is we want to invite everyone who wants to get involved. If you go to fightcancer.org/NYBio, so fightcancer.org/NYBio, that is the homepage for our biomarker testing campaign in New York. You'll be able to, if doctors are invited to sign on to a letter to lawmakers, patients, and everyone who cares about this are invited to, join the campaign and get added to our listserv. So we'll keep you informed. We're looking for patient stories. Just go to the page, you can contact me and get involved. So the last thing I just want to ask Dr. Morrison, I'm going to put this question to you because I heard you once before say how this was a fascinating question. What does the future of biomarker testing look like? So we've got 2 minutes left. So in 2 minutes, how would you summarize what's the future of biomarker testing look like?
I think it's two things. It's accessibility, and I mean from a perspective away from test to doing all testing in blood or so-called liquid biopsy that you could walk into Rite Aid or CVS or think that maybe 10 years from now you just walk into the grocery store and have your blood drawn, have your biomarker testing done. I really think technology has changed. We will be doing testing routinely from blood for all tumor types and all disease types in just a matter of another year. So I think the other point that goes to that is early cancer. Right? That's where we're moving toward. Right, it's tech disease. It's early stage when it's more readily cured. Thank you.
Excellent. Thank you so much, Dr. Morrison. Thank you, Tiffany. Thank you, Dr. Swat. And thank you all for participating today. We really appreciate it.
Thank you.
Thank you for joining us for NewYorkBIO's Patient Engagement Summit, and to the patients, partners, and sponsors that help make this event possible. Please visit our website at www.newyorkbio.org and follow us on LinkedIn and Twitter for more.